Hey everyone, welcome to our chapter 4, which is all about cells, and lab 5, which is all about biochemistry recap video. So let's go ahead and get ourselves started. So, in chapter 4, the very first thing that we talk about is cell theory, and there are several different scientists that are involved in discovering and sort of writing down and quantifying what we know about cells. And from all of their research, we find really kind of three major parts, although two of them sort of go together. The first is that all living things are made of cells, which means if it's got cells, it's alive. If it doesn't have cells, it is not a living thing. And this is the part of cell theory that really has scientists struggling with classifying things like viruses that don't have cells but do reproduce. And so it makes it sort of a research focus area in biology. But our first part, again, is all living things are made of cells. And that cells are the structural and functional unit of living things. What that means is that if we break cells into their smaller parts, it's not alive. It doesn't do the things that living things do, like reproduce and develop and have a metabolism. So that cell is the smallest functional unit of life. And then last but not least, cells come from other cells. There's no spontaneous generation. They don't poof into existence. It's really just mitosis or meiosis, this process of cells reproducing to make more cells. And then the chapter goes into prokaryotic cells, which are our more simple type of cell. It includes the domains and kingdoms of bacteria and archaea, respectively. We can name these cells based on their morphology or their shape. Bacillus is the kind that looks sort of like a tic tac, or they say it's rod shaped. That's this big fuzzy green guy and this little pink string. We also have coccus or cocci, which are circle, and spirillum, which look like little spirals. The components of prokaryotic cells are the envelope. It's also sometimes called a glycocalyx. It's made of this sugary substance on the outside. Then we have a peptidoglycan cell wall. That cell wall, again, made of that protein sugar base, protects then the plasma membrane. And in our big picture, it's like the envelope is the lime green, the wall is this purple or red-ish line, and then the sort of teal green would be the plasma membrane. Sometimes outside of our prokaryotic cells, we have fimbriae. They're sort of like Velcro projections to help stick the bacteria or archaea in place or to each other. There's also pili, which are like little straws or tubes that allow for reproduction, where we can give DNA to a different prokaryotic cell. Then inside of that plasma membrane, we will have the cytoplasm, which is the goopy stuff with all of the floating around components. We will have a nucleoid region that contains the one circular chromosome chunk of DNA. That is this big red thing on our diagram here. Sometimes we have plasmids, which are smaller chunks of DNA that are sort of like an emergency backup system in case our cell finds itself in new or harsh conditions, that maybe that little chunk of DNA has the code to help it survive. We also will have ribosomes. They are a little bit different in structure than our eukaryotic ribosomes, but they do help to make proteins for our prokaryotic cells. Sometimes we'll have flagella, which are like tails that whip back and forth to move our prokaryotic cells. But really, that is about it when we're looking at these more simple prokaryotic type cells. Which then we need to talk about how we get to our more complex eukaryotic cells. And your chapter gives you endosymbiotic theory, which says that over time, modifications to prokaryotic cells have given us our eukaryotic cells. So, what does that look like? Well, if we start here at this little wavy timeline, we've got our prokaryotic cell with its DNA, with its cytoplasm, with its plasma membrane. And over time, that cell grows and gets bigger and bigger. Um, but then it's sort of too big to function well. And it starts to have this infolding, bending, folding of the plasma membrane. And we create these little compartments. And we're going to see that we can call those compartments different organelles like endoplasmic reticulum, nuclear envelope. It starts to make areas to have functions. And then we can have a ancestral eukaryotic cell live in its life, and maybe it engulfs a different type of prokaryotic cell that can make energy. And it doesn't dissolve and destroy that because it's been useful to it. And so after a while, it sticks around and now it's in all of our eukaryotic cells. And then maybe a little bit later in this timeline, we have this eukaryotic cell that can engulf a prokaryotic cell that does photosynthesis, makes its own food, and it doesn't dissolve and destroy it, but again holds onto it because it's useful. Now, there is evidence to support this endosymbiotic theory, one of them being that the plasma membrane that is found in those organelles inside the cell has the same phospholipids and proteins and sugars that we find in the membrane outside of our cells. If we look at mitochondria and chloroplasts, we see that they do have their own DNA, and that they have a sort of bilayer of membrane. So it's like they have their own membrane, and that something brought them in. And we see that. So that is how we get to this endosymbiotic theory. That being said, if you're very interested in, there are other ideas out there. This is just the one with the evidence that is most supported. So it is the one that we learn about. Which then leads us to our eukaryotic cells. We have plant cells, the green square one over here, and we have animal cells represented as this blue round one. It is good to note that these are our representative cells, and that the cells you actually find in plants and actually find in your body, they could have all of these organelles. They could have some of these organelles. They could look different than these representative models that they show all the things. Some things that we share with plants is that there is a membrane-bound nucleus. It's purple in this plant and pink in this animal cell, with a nucleolus responsible for making our ribosomes. That's the little golf ball inside of our nucleus. We then share having endoplasmic reticulum. That's right outside of this nucleus in green on the animal cell and blue on the plant cell. That is a place where ribosomes can be attached, and we will make proteins and package them up. Or it's the smooth endoplasmic reticulum where there are no ribosomes attached and we modify fats. We will see that there are mitochondria. It's these jelly bean with squiggle lines in pink and yellow, and then red and pink in the plant cell. That is our powerhouse of the cell. So to say, it makes our ATP energy source. Um, we will see also a Golgi body or Golgi apparatus. That's this orange and orange as well in the plant. That's like our UPS. It ships different products in and out of our cell. We've got our little straight line looking things. That's part of our cytoskeleton that is going to be holding the shape of our cell. A few things that are a little bit different is that in our animal cell here, we've got a plasma membrane, and it is made of phospholipids. In our plant cell, we have a plasma membrane. It's this light green, made of phospholipids, but then we have a cell wall. And that cell wall is going to be made of cellulose, and to make structure for our plants, it helps them be able to grow like a tall tree, right? A human can't be as tall as a tree. Our cells don't have that rigid structure. We also see a difference here in the plant cells. These green things that have little stacks of Oreos in them, those are chloroplasts. That's the organelle responsible for photosynthesis. In animal cells, we have these centrioles. Centrioles are going to organize our cell when it's time to divide in mitosis. It makes the little spindle strings that will pull our chromosomes apart. Over here in our plant cell, we have this big bubble that is called the central vacuole. It holds sugar and water, sometimes pigments, or food sources, and that is usually not found in eukaryotic cells. When they have a vacuole, it's mostly to get rid of water. We call it a contractile vacuole, but that's not very common. It's not even represented on our animal cell picture here. If you are hoping to get a little more depth and detail, I highly suggest and recommend the Amoeba Sisters cell tour video. It's about 10 minutes long. I think it's like 9:38. It's very awesome, and they give you an analogy of our cells to parts of a city, which is usually very helpful for students. It is found in the extra resources tab. I think that that is a fantastic way to learn the cell organelles. And then, whether or not you watch that video, get you some bonus points. Feel free to email me answering the following three questions. Name and state the job of three organelles in both animal and plant cells. So you can choose whatever three organelles. Then name one and state the job of that one organelle that's only found in animal cells. And there are a few things only found in animal cells, but just pick one. And then name one and state the job of that one organelle only found in plant cells. And again, there's more than one, but feel free to just tell me one. Email me that, and we'll put some bonus points on to your grade. Then our lab is all about biomolecules. Now, what is that? Think of these as the big molecules that will make up the components of cells. That's going to include carbohydrates, nucleic acids, proteins, and lipids. So we are going to use these big molecules to build the cell organelles and make our cells. In order to build these big molecules, we need smaller molecules to make them. So they are made of monomers. Think of that like one Lego building block. And we join those monomers together to build polymers. Polymers are the big molecule. Think like an entire Lego house built of the Lego bricks. In order to put them together, the chemical reaction is called dehydration synthesis. We remove the water, and we now have two monomers stuck together to start building our polymer. If we want to take that Lego house apart, all we have to do is add water. We call it hydrolysis. Add water to our polymers, and we will break them into our monomers. In your virtual lab, you will be testing several different um, substances in order to determine if they have a biomolecule present. So you're going to use Benedict's test. Benedict's is a blue chemical reagent that when in the presence of monosaccharides and disaccharides, the monomers to carbohydrates, it will turn sort of a yellow to red, orangey color depending on the concentration. We test iodine. That is a yellowy brown, I say amber. Some people call it Dijon mustard colored chemical reagent that when it is tested against starch, which is the polymer or big molecule for carbohydrates, it turns kind of this bluey black dark color. You have Tween as an emulsifier that is going to be able to detect liquids. Tween is clear, but when we mix a water and a fat, a lipid together, it becomes cloudy. There's also the paper drop test where you put a drop of a substance onto brown paper bag or paper towel and let it dry. And if it leaves a residue, a spot, then you know you have a fat, a lipid. If it doesn't, then you do not. You have done this experiment in your life. If you've ever eaten fast food, like say McDonald's, and they give you your McDonald's bag and you see the greasy stain from the French fries, and you're like, oh man, my French fries spilled in the bag. You've already done this paper test. Then there's Biuret, which is a blue um, chemical reagent that, when mixed with proteins, turns pink or purple. And you will do virtual labs to test these different chemical reagents. So then last but not least here is looking at sort of our schedule. Don't forget today is the due date for exam 1 and the work from last week. If you need more time, please email me before the end of the day today, and I'm happy to get that updated for you. Then we're moving ourselves forward to Chapter 4 with the Chapter 4 Smart Book, Virtual Lab 5 with those modules. There is a lab quiz that covers Lab 4 from last week and Lab 5. Wednesday is just a continuation of, the Chapter 4 and Lab 5. Thursday, we move ourselves forward to Chapter 5 and Virtual Lab 6, and we will have another video that walks you through that. And then I like to say our Friday, Saturday, Sunday is our catch-up, must-do, and mayo. Catch up any work from week 2, like our Unit 1 discussion board, our smart books, anything like that. You must do, that is your week 3 work with your smart books from Chapter 4 and 5, with your virtual lab modules from 5 and 6, and of course, those quizzes. And then, of course, our Mayo or May do, you can work ahead. We will keep it moving to Chapter 6 and Lab 7 and Exam 2 next week. All right, thank you for watching. And of course, let me know if you have any questions at any point at our material we're working over. All right, we'll be in touch soon. Bye.